I'd like to show some of the work we've been doing recently on a technique for label-free biomolecular tissue analysis at arbitrary depth. This will be mostly in the mouse brain, so I understand this is not the exact model you're all working on, but in perspective, it might be something that is interesting for all of you. So I'm a physicist. I'm here at the Department of Physics in the Padua Neuroscience Center. So this will be the take of the presentation, and I'll be more than happy if you'd like to discuss about something in the following. So as a brief introduction, as I'm sure you're all familiar with, in the last couple of decades, there's been a lot of advancement in the study of optical interfaces in order to control and monitor neural activity, mostly in the mouse, thanks to the availability of genetically encoded molecular actuators and reporters that allow to inhibit activity, stimulate activity, or to read out activity. Now, if you look at the probes, you need to do this. There's two main kinds. The one on your right that are that are based on very thin chunks, for example, on silicon, where people put on micro LEDs, recording electrodes, or photodetectors, all sorts of photonic application that allows you to have an optical interface with the brain. On the other side, you have multimode optical fibers, where you have a fairly standard fiber optic that you can place in a moving animal and go and stimulate or record the activity. And I will mostly focus on this family of probes. What is interesting is that these allow you to go much deeper than what you could reach, for example, with a 2-photon or 3-photon microscope, where you're limited to maybe 1.5 millimeters if you're very good. Here you can go down to 3 or 4 millimeters. So there are a lot of development in fiber-based neural interfaces. So the first one, you have the fiber photometry. What is fiber photometry? You have a mouse doing some behavior, and you use a multimode fiber, for example, to send light and collect path light. This can give you information, for example, of calcium activity through a genetically encoded reporters, and you can link the function to the movement of the animals. Then you can scale up the complexity in order to have, for example, depth resolution, where we have been looking at dopamine transients when a mouse was running for a reward in the striatum. You can have multiple fibers alongside recordings, and you can also have some endoscopic imaging with subcellular resolution with 100 microns diameter of the fiber. So all of this is based on fluorescence. And what we do is we measure variations in space and time of fluorescence, and we link it to neural activity. However, there's a lot of information that we are missing, because what is not encoded by the reporter, we are not measuring. For example, if we have alteration in the site architecture, imagine traumatic brain injury, we're not measuring it. If we have a loss of myelin that is not reported by the probe, we are not measuring it. So there's a lot of information that we are missing with the state-of-the-art technology. So what would be interesting is to have a biomolecular point of view on the brain mechanism that goes beyond the genetically encoded reporters and ideally does not need a reporter, so that is label-free and you can measure the brain in its native state. So one way to do this is to look beyond fluorescence. So in fluorescence, if you take a physics perspective, what you have is you have a fluorophore that absorbs and emits light. So you deliver some light that is absorbed by the fluorophore, for example in the blue, and then you have your green response and you measure that. However, this is not the only phenomenon that takes place in the brain. When you shine light on any kind of element, but also on biological tissue, you have another effect. It is Raman scattering. In this case, your light is not absorbed, it is diffused by the molecules you are scattering against, but some of the energy of the light is lost to the vibration of the molecule. So the molecule vibrates and steals away some of the energy of your light, and you get light at a slightly different color. The interesting thing is that the color you measure gives you a fingerprint of the vibration you have scattered against. So you can have a biomolecular fingerprinting, label-free and non-destructively, of your biological tissue. This is very interesting. The drawback is this is infinitely weak. So 1 in about 10 million photons does Raman scattering. So you need strong intensity, long acquisition time. But the paycheck is that you have label-free information. For example, here an example of olive oil and ethanol, and you have molecular information from your sample. This technique has been widely employed, and one of the most interesting applications is its use in intraoperative Raman spectroscopy. Picture the case of a surgeon that has to remove a tumor from the brain. He does the surgery and is left with the surgical cavity. Now, the amount of tumor that she has removed is fundamental for the prognosis. So if no malignant cell is left, the patient has much better possibility to survive. So very recently, led by the group of Leblond, people have developed probes that can be handheld and scanned in the surgical cavity, and using the Raman spectroscopy can understand if some malignant cells were left in the operation, and then go forward and remove more. And this is very interesting. Now, it would be fantastic to use this same capability also in the mouse to see if a biomolecular alteration is linked to a functional alteration, so that we can then establish a bridge between the two. The problem is the scale, because these probes are pretty big. We are talking about 1 or 2 millimeters, but the mouse brain is small. This is a rough comparison in size. So remember, the Raman signal is very weak. To collect a lot, you need a large lens, basically. But if you have to shrink down to the mouse brain, you cannot put a large lens in the brain. So this is the technical challenge we have tackled, and we've done it using a particular case of optical fiber with a tapered profile in order to introduce an approach for vibrational fiber photometry. Vibrational is a short word for Raman spectroscopy that can be added to the standard fiber photometry I've described at the beginning. This is advantageous because you can get at once information on the site architecture, on biomarker, or on the molecular composition of the tissue that surrounds your probe. Because the properties of this particular fiber is that instead of emitting only at the end, you can use a cone to deliver light and to collect it back into your probe. And this allows you to go and recruit information from about 1 millimeter of tissue in depth. And in the case of the mouse, this is a lot. Consider that it is about a factor 5 or 10 with respect to what you can do with standard applications. So here's an example. We are shining light in a slice, fixed slice of the mouse cortex, and you see this is all the region of tissue we can recruit for. It's basically all the cortical layers. So to which use? The first one is extracting region-specific spectral signatures. So you see on the left a slice where we have done the measurement in the whole brain and then performing the sectioning in collaboration with the KSIC. And here is the footprint of the probe. As you can see, it is very small. You can barely pick it out. And if you look at the spectra extracted from the cortex, the hippocampus, or the thalamus, you see that there are spectral differences. These are due to the different content of protein and lipidic content. Another interesting example is the use in the spectral characterization of tumor formation, for example, melanoma brain metastasis, where you can see that if you measure in the metastasis or in healthy tissue, you have different spectral signatures, for example, the presence of melanin or a diminishment in the lipidic band and in lipidic and protein band that has very good diagnostic accuracy. Now, I'm pressed for time, so I'll skip a few things. These methods also work in vivo. This is an example of in vivo characterization of the brain metastasis, for which we have developed a dedicated system that can be fit in a standard stereotactic rig. And it can also detect more nuanced molecular alteration. Of course, a metastasis is a very clear and confined difference, but the case of traumatic brain injury can be a bit difficult to pick out. But if you look at the spectra extracted from healthy brain or in the lesion, healthy is the cyan and the lesion is the magenta, you see that compared to histology, we can pick out the loss of myelin in the lipidic band, the nuclei loss in the protein band, some sign of the rupture of the blood-brain barrier, and also some weak signal that we think could be linked to the presence of inflammation in the Hoover number linked to phenylalanine. So overall, this allows you to detect quite a wide range of variation in the composition of the tissue, and the idea is to couple the possibility of label-free optical molecular analysis together with functional recordings, and then jump to the behavior. So this is a few years' work for now. If you know anyone that might be interested in helping out, there will be open positions, so please feel free to share the information. Let me then finish by acknowledging all the many people that have worked at the University of Padua, Italian Institute of Technology, the CNIO with Manuel Valiente, who has developed the metastasis, Lisette Della Prida at K6, and other people you've seen listed here. So thank you very much for your attention. I'll be happy to take your questions if you have.